So you could make an egg develop into a normal organism with an older nucleus than what an egg normally would have. So what he was more, so he did that experiment, and that was promising. But he actually, in 1938, so 10 years later, in a book called Embryonic Development and Induction, he made a prediction that, and his prediction was that there would be a fantastical experiment where you could actually clone something, and so he actually coined this word cloning, by transferring a nucleus from a differentiated cell into an enucleated egg. So he's the first one who said, I want to try this from a differentiated, meaning an older, presumably even could be adult cell. And he never did it, and he actually didn't think it would work, but he thought it would be amazing if you could. Because if you could take a cell from an adult tissue that has its own fate and cause that nucleus from that cell to trigger all of embryonic development, then it proves that our genomes are not actually altered as we differentiate. It just shows that they are regulated in a different way. So following on Spemann's work were two scientists, Briggs and King, and they in '52 did a really landmark experiment, which was somatic cell nuclear transfer. And before I tell you exactly about their experiment, I just want to mention Briggs because he seems like a character. So he worked in a shoe factory and earned money as a banjo player before becoming a scientist. And then once he did actually become a scientist and get his own lab, he applied for a grant to study this process, nuclear transfer. It was immediately rejected, and they called it a harebrained scheme with little chance of success. So I think to any of us who've gotten grants rejected, we can sort of commiserate with him. But he did get money eventually and hired King right away, who had been trained in